Hello, folks. This is going to be lecture, class 30 about recombination and various topics related to it. As soon as I get a chance to share the screen here with you. All right, so again, class 30, in this case, due to a snow cancellation of in-person classes, or this might be reused in the future. So the topic is recombination, chromosomal alterations, as well as somatic recombination. A specific example we'll talk about related to the immune system for the somatic recombination. So the goals today are to look at recombination, the process that we've often referred to as crossing over that happens during meiosis in the formation of sperm and egg. Or it could happen in other systems where sperm and egg aren't the outcome, but maybe the ovule and a sperm-like sort of structure that houses a genome from one of the sexes of, for instance, a plant. But we will look in this case, we'll be looking at how recombination happens in detail, something we haven't looked at previously. Then we'll talk about how chromosomes can get kind of messed up during this process, and we'll end it with a discussion of this topic of somatic recombination that occurs during antibody formation in B cells. And I think to some extent, this will be covered perhaps even in greater detail if you take the immunology class here at Augsburg. So let's talk about the role of recombination in meiosis. Recall from previous studies that this process of meiosis is a type of reductive cell division whose goal it is to reduce the chromosome copy number. So we start out at the beginning of meiosis here. The cell has just duplicated its chromosome, so it has 2 copies of every chromosome. This model example shown here has 3 total chromosomes, a small one, a medium one, and a large one. And the red copy would be, for instance, from the egg parent, and the blue copy, the sperm parent. So in this case, we have a diploid cell at this stage because, again, the chromosome copying happened just before meiosis started. We go through the multiple rounds of meiosis, the multiple steps of meiosis 1. That's the first stage. And we will, in a minute, focus on what happens in early prophase of meiosis 1. That's when crossing over happens. Then recall that the chromosomes the nuclear membrane breaks down, the centrosomes, these microtubule organizing complexes, go to the poles, and they send out 3 populations of microtubules: the aster microtubules that keep the centrosome positioned, the kinetochore microtubules that directly connect to the chromosomes, and the polar microtubules that kind of span the whole distance and keep the 2 centrosomes kind of positioned. And in the center, we see what's called the metaphase plate. Once we get to metaphase, where all the chromosomes line up. Remember that the red and blue chromosomes assort independently. That is, they don't have all red on one side, all blue on the other. Notice that here in early prophase, it's showing late prophase, but in early prophase, the recombination or crossing over happened. And that shows up here. So we see a blue chromosome with a little bit of a red tip, a red chromosome with a blue tip. And as these cells then go through meiosis 1, half the chromosomes, one of each variety, small, medium, and large size, small, medium, and large size, end up in each new cell. Each of those cells then undergoes meiosis 2, same steps, except there's no recombination or crossing over in early prophase of meiosis 2. Just the movement of the chromosomes, alignment on the metaphase plate, and then in this case, the sister chromatids, or the monads, are pulled apart. The end result is 4 cells that each have just a single copy of each of the 3 chromosomes in this case. So that's a quick overview of meiosis 1 and 2. And again, the recombination part that we're going to focus on happens right here in this stage I'm circling. Actually, this says late prophase. It starts in early prophase. So again, the phases of meiosis: early prophase 1, late prophase 1, prometaphase, metaphase, anaphase, telophase, and cytokinesis. And the prophase of meiosis 1 has these early steps: leptotina, zygotina, pachytina, diplotina, and diakinesis. I don't expect you would memorize those. I don't have them memorized. But I do know that early, in one of the early steps, the crossing over of chromosomes begins. it forms what's called a bivalent. You can see it here. That it's sort of like a zipper. So this jacket I'm wearing, I could zipper up the neck area here. Okay, so think about the two chromosomes kind of coming together and zippering up like my jacket. That junction between the two chromosomes where they start to connect is called the synaptonemal complex. We've looked at this previously. We looked at some of the evidence for it. There's electron micrographs that show it. We know about the proteins that are involved in helping bridge the chromatids here. So it's not the entire chromosome. It's just in this case, the inner two sister chromatids that form this complex. And then they create a crossing over. So the union of those two sister chromatids is called, the junction is called the synaptonemal complex. We refer to it as a bivalent. Two items are coming together, and then they swap genetic information. So the chromosomes are actually cut and they are reassembled with, as you can see here, this chiasma. So they stay kind of connected. They've given each other a little bit of information. But even into the end of this prophase 1, you still see them connected. So that's what we see right here, the late prophase. But again, the steps begin in early prophase. And then finally they separate. And once they separate, you can see here down at the bottom right, the end of prophase 1, we now have chromosomes that are sort of hybrids. They have some of the blue and some of the red, most of the red here and a little bit of blue. So here's just a little bit of red on the blue chromosome. And so that's the material that was exchanged between the two in this process we call crossing over, or more technically correct, we call it recombination, homologous recombination. Okay, so how does it happen? Just reviewing again from a previous lecture several weeks ago where we looked at how it is that the chromosomes kind of come together. They find an area of similarity. And again, they're going to be highly similar because they are homologs. That is, one is the version of that chromosome from one of the parents and the other is the other parents. So there will be areas where there could be differences, but overall they will be highly, highly similar. Synapsis occurs to form the bivalent. And then in these examples, we just show one crossing over, but crossing over can occur multiple times within a given chromosome. So here we see two crossing over events between this red chromatid and the blue one. And at the end of this, the information that's exchanged is actually between parts of the original chromosome, which is all red or all blue. And so now we've got red with a blue chunk inserted and then red. And the blue chunk is just swapped with this red piece here. So the chromosome exchanged this area between the two pairs. How does this work? People had seen electron micrographs indicating that there were kind of these crosses made and this concept of the chiasma. You can see that on electron microscope images, like transmission electron micrographs of stained chromosomal preps from cells that are actively undergoing either mitosis or meiosis. So in this case, it would be meiosis because of crossing over. And what scientists came up with is several models. So the prevailing model is called the Holliday model. It was proposed by a British scientist, Robin Holliday, sometime in, I believe, in the 70s. It might have started in the 60s. And we're not going to go into detail about a lot of models. This is just kind of presenting you some ideas. So there are at least two other competing models. And I'll talk about the relative strengths and disadvantages of this Holliday model. But the basic idea here is that you have two almost identical chromosomes, right? They're homologs. So one came from the egg parent, one from the sperm parent, originally. And so they do have allelic differences. That is to say that the genotype of this particular chromosome is big A, big B, and the genotype of this one is little a, little b. Okay, so during meiosis, part of the point of meiosis, kind of from an evolutionary perspective, is that it allows an organism some flexibility in rearranging the alleles, parts of genes that are shared, that are maybe highly conserved, but there can be allelic differences. And the genotypes, big A and little a, are different alleles of the A gene. And so in terms of what advantage this provides to organisms, they can make sperm and egg that will then create new organisms that have a different combination of these A's and B's. So let's look at this. This would be kind of a classic true breeding line experiment in the sense that we've got a homozygote dominant chromosome with big A, big B, and a homozygote recessive with little a, little b. But the process of sperm and egg formation in this organism will result in kind of a shuffling of these big A's, big B's, little b's. So that's what we'll look at. So Holliday proposed kind of an outlandish idea that at an identical position on both of these blue and pink purple chromosomes, there will be a break of the DNA, okay? So breakage, sometimes called NICS, is fairly typical. It can happen for a variety of reasons. There could be an enzyme, there could be some kind of chemical damage. You could have mechanical sort of torsion that would separate the DNA. And so the idea that's the reason this model has, this is the main weakness, is that the NICs have to be in the exact same position on each of these two separate chromosomes. So, but let's just assume that this could happen. Okay, so the NICs occur in both the blue and the pink strands. The sister chromatids here are the darker blue and the lighter purple, or the pink strands. Notice that the outer strands of the double-stranded DNA are not affected. So again, crossing over is just occurring between we could think of it as the inner sister chromatids. They're not really inner or outer, but the two that are closest to each other. Okay, that's where the pairing up first happens. The bivalent forms. We get this chiasma, this kind of X-shaped crossing over thing. And then ultimately there's an exchange of genetic information. All right, so we get the double-stranded break. These two chromatids are close to each other. There's a breakage of the DNA. Now what happens? Well, once the NICs are there, the tendency is to fix them. And part of that process might be, but things are happening kind of rapidly in the cell. So I like to think about, moving your fingers like this kind of repeatedly. Imagine inside of a cell, all the molecules are moving around. They're in constant flux. Depending on the temperature of the system, they would move faster. If it's colder, they would move slower. At body temperature, things like, if we think of this as being a mouse or a human that likes things, somewhere in the 90-degree range for the body temp, 98.6 for humans, Fahrenheit. Is that true? Yes, that's the correct temp. So 37 Celsius roughly. There's quite a bit of thermal sort of energy in the system, so molecules are constantly moving. So imagine the two DNA strands break, and they're sort of doing a dance, and they bump into each other. And then we get what's called strand invasion. So this part of the blue strand can pair with the dark purple strand. I mean, it's homologous. So we get this thing called a Holliday junction. This was what Robin Holliday discovered. This is the point at which the nicked positions on the DNA allow for the strands to kind of cross over and connect. And then we can get the reformation of with DNA ligase is involved, a reformation of the double-stranded, but now they're intertwined. And so there's kind of an X position where the two different chromatids are crossing over, and they form this structure that under electron microscope looks like the Greek letter chi, C-H-I. Some people pronounce it chi, but it's sort of an X-shaped symbol. And that's what this looks like under a transmission electron microscope. So Robin Holliday and others called it others called it the Holliday junction, but it's like a chi formation. Okay. Not to be confused with the statistical test, the chi-square test, but this is just the look of it. It looks like an X, okay? So once that's in place, this is an unnatural thing for the cell. The chromosomes normally shouldn't be connected like this. It just happens during this special time in meiosis. And the next step is that there's nothing preventing these strands from kind of tiling along left or right. That is to say, the hydrogen bonds are pretty easy to break. And it's under a little torsion here because you've got this crossed over thing, and now there are two molecules that can twist potentially. So there's something that's called branch migration. So we think of this as a branch point, and imagine that this junction could slide to the right. you would just require that the hydrogen bonds here be broken and the ones in the back behind this chi formation, that they reform. So that's what we see here. Notice that this junction now has slid to the right, so it's over here now. And so all of these base pairs had to be broken, and these had to reform in the wake of the moving junction, which kind of goes from left here to the right. Okay, that's what you see here. So these are called heteroduplexes now because hetero means different. And so duplex two, so we've got two chromosomes that are hooked up together and they've exchanged some of their respective information, which is again, slightly different variations on the same sequence. That is to say, little a, little b is different than big A, big B, but overall the genes that those differences are in are highly similar, okay? However, once the pink or purple chromosome here exchanges information with the blue chromosome, then we have these hybrid chromosomes. So the heteroduplex region refers to this area where you've got the light blue, which was the original sequence. Then we have some of the original dark blue sequence up to where it broke. Then we have this new sequence, which came from the other strand over here. And then at the end, we see some more of the dark blue sequence, which was the original sequence. So the combination of the original light blue and this new pink sequence is what we call the heteroduplex region. And you see it on the other side as well. So there may not be complete perfect matches here if there are slight differences in the sequence between, say, the big A and the little a gene, or the big B and little b gene. Okay, so once the branch migrates from left to right, the direction of this arrow, now the thing of it is I'm in my kitchen, so I'm going to use knives, kitchen knives. So we've got two kitchen knives. They're sort of parallel to each other, right? And so the branch is migrating. Is this going to work? Yes. And so they're parallel. You can see the knives are lined up. Now, and there's a slight area of connection. But what about the knives twisting? Okay, so let's look at that. Around this X junction that I'm circling with my cursor here, there's nothing that prevents these two knives from kind of twisting like this. Okay, kind of like if you were standing and you can scissor your legs, right? And so that's because remember, again, this is inside the nucleus. There is thermal energy. Molecules are bumping around. They're in kind of constant motion. And so they're not always going to stay aligned parallelly. In parallel, they can move like this, just like these knives. So the chromosomes, as they twist, that could change the connections a little bit. So let's take a look at the migrating branch. And so here's the migrating branch. And we're going to think of two things as happening, okay? So this area of kind of the wiggles would be the heteroduplex area. We note that the outer branches here have big A, big B, little a, little b. But in the middle, we would have little a, little b, except there's been some exchange of information, okay? So that's how we get the kind of shuffling of the genetic deck of cards, is that during this crossing over, each of the chromosomes can contribute their own alleles, and we can end up with these hybrid chromosomes at the end in the sperm and the egg at the end of meiosis. So here's the thing. I don't think the knives will work for this, but the basic idea is we can either twist it this way or we can twist it kind of this way, okay? And so that's what we're doing in this example. So the carryover, it looks like this. And in this case, we're just going to cut it and then form the two new chromosomes. So if we just did a cut right here at this junction, and that's shown here, then this strand would be some of the old dark blue, the new light pink, and the old dark blue. So this middle sequence, this heteroduplex, would be coming from the original chromosome here, but now it's going to be part of the blue chromosome. Okay, so that's what happens, and we end up with the same alleles. So all that happened there was between the big A, big B, little b allele, in the middle of it, some new sequence appears. But because we are only looking for the traits that are specified by big A and big B, unless we sequence the DNA, we wouldn't even know that there was this new intervening sequence. It could change the function of the protein slightly, we don't know. But the point is that even though there was crossing over here, we we don't really detect it in a typical phenotype screen, perhaps, because there's just a short region of new information that doesn't seem to change the alleles significantly. Now, what happens if instead of, again, we'll go back to the knives example. So we had the two knives were lined up, there was some crossing over, and we resolved it. But what if instead of doing that, we twist these things a little? And so that's what happens here. So we see this crossed over region. Now imagine, it's a little hard to imagine, but grab the top strand with your left hand, the big A, big B, and grab the bottom strand, little a, little b, the purple strand, with your right hand, and then rotate just the one hand. And you unwind this, and that's what we see here, okay? So we still have these heteroduplex regions. We can see the original little big A, big B is shown here. The original little a, little b is now down here. And then here we've got some of the original dark blue chromosome, the heteroduplex, dark blue chromosome, heteroduplex. And so now imagine a scenario where this gets cut in this dimension. So instead of cutting it here, we're going to cut it here. And now if we resolve that, ligase has to come in and link this purple to this light blue to this purple, and ligase or another enzyme would have to link this up. And so what we get is big A, little b, big B, little a. So we have, and if we go over here, we can see that the outer sister chromatids are totally the same, big A, big B, little b, but the inner ones had this piece exchanged, and now that exchange affects the actual alleles. And so we get little a, big B, big A, little b in those inner chromatids that exchanged information. So the Holiday model describes all of this. We can map enzymes onto this. I'm not kind of going into that detail in this lecture, but there are a whole system of enzymes that are known to be involved in homologous recombination. And some of those overlap with other functions in the cell, just like we've seen before. So we have to have nicking enzymes that cut the DNA. There are some enzymes that help with the strand migration, the strand invasion, if you want to call it that, the kind of formation of the Holiday junction. There are other enzymes that assist in the migration of this branch. And then there are enzymes that have nickase activity or endonuclease activity that do the cuts like this that we see here, where the DNA is cut, and then ligase has to put the DNA back together. So there's a whole system that does recombination. And a lot of our understanding of recombination comes from finding mutants, particularly in E. coli, that are deficient in some aspect of recombination. Okay. So there are a variety of videos I identified that are quite good at going through the concepts of homologous recombination. And so I would encourage you to watch those. We're going to shift gears now. This is the second topic for this particular class. And we're going to focus on things that can go wrong with chromosomes in the cell. This could happen at really pretty much any time. But we think of the processes where the chromosomes are unwound and kind of interacting as being the most vulnerable. And so most of this we would think of during mitosis or meiosis, as there's recombination occurring. So we talked about recombination in meiosis now and sort of restricted it to that and gave you the impression that it doesn't happen during mitosis. It's very rare in mitosis. It doesn't happen in all types of cells, but it can happen even in mitosis. There can be recombination. But it's more prominent and sort of regular, let's say, during meiosis. Okay, so let's just talk about this. So during mitosis, there can be recombination between chromosomes that is inappropriate, that can fundamentally alter chromosomes. That's called mitotic recombination. There are examples in fruit flies and humans as well. As long as there's no DNA lost, there will be probably little effect, although we'll talk about position effects a little later, because those can lead to some alterations, and we can see some human diseases where this happens. So this seems like a lot. This is basically a slide for you to kind of organize your thinking about this concept of changes in chromosomal structure. We can have deletions, oftentimes called deficiencies. That's where a chunk of chromosome is permanently lost. It could be at the end, it could be in the middle. Then we can have duplication, where during the process of usually DNA copying, but also it could happen during recombination, you get an area that gets doubled. And so you have twice as many genes for that region. Inversion is where a chromosome just sort of flips its information. It's still all there, it's just in the opposite orientation. And then translocation is where chromosomes swap parts. We don't think of it as recombination because it's due to a different system of enzymes called transposons and transposases. Sorry, that's not true. It could be due to that, but mostly this is just inappropriate crossing over that should not happen. It could be simple where part of a chromosome just goes to another, or it could be reciprocal where they each give each other a little bit of the DNA from their chromosome. So these are the four major ways that DNA can be altered, the chromosomes. Here is a graphic cartoon-esque kind of view of that. So here's a deletion. You can count the numbers. We've got 4, 3, 2, 1, and now we've got 4, 3, 1. Okay, so the 2 was deleted. This whole region between the red arrows was lost. That's a deletion. A duplication, 4, 3, 2, 1, 4, 3, 2, 3, 2. So we see 4, 3, 2, 1, but the 3, 2 was somehow duplicated in here. What about inversions? Again, we're reading from left to right here, 4, 3, 2, 1. This is the center or centromere of the chromosome. Then on the other side, it goes 1, 2, 3. But just look at what happens between these arrows. We're not going to lose the information, but the 3 and the 2 kind of split, okay? And so then we end up with 4, 2, 3, 1. The numbers here simply represent fictional kind of markers on the chromosome so that we can study these alterations. Okay, so translocation, a simple translocation, the blue part of this tiny chromosome 21 is going to translocate into this region. So what we see is, that's not what's going on. What is going on? So this region between 4 and 1 is going to move into this chromosome with the blue at the end of it. That's called a simple translocation. It's unidirectional, in one direction. So this part ends up on the end of the purple. You can see it 4, 3, 2, 4, 3, 2 between the red arrows. And then it tacks on to this 2, 1 and makes a new chromosome. The other one lost some material. Now, that's a simple one-way translocation, but what about reciprocal translocations? Well, that's where each chromosome gives the other part of itself. And so in this case, the 4, 3, 2 will go over here, but this one is also going to send a little bit of this blue over here. Not all of it. You see some of it still here, but the part kind of from the black line to the centromere, you can see right here, ends up on this chromosome. Okay. So now we're going to go through each of these types of alterations of chromosome in turn, looking at examples in many cases of them. One big question is, is it damaging to delete part of a chromosome? So the answer is it depends. It depends on the size of the deletion. If it's a big chunk, it's more likely to have an effect. It also depends on what's in that region that is lost. Are they critical genes for the function of cells? When are they critical? If it's lost later in life, would that be less damaging than if it was earlier, etc.? Okay, but in most cases, we're talking about the formation of sperm and egg. So a deletion could be pretty damaging if it hit a significant gene or took out a big enough chunk of the chromosome. Okay, so when deletions have a phenotypic effect, they're usually detrimental. That is, if you can see it in the phenotype, it's probably an issue. We're going to look at the example of cri du chat in humans. This is a French phrase for cry of the cat. So in some ways, it's a bit pejorative because it's describing the phenotype of the newborn that is known to have a high-pitched cry, not unlike that of a cat. And so that's how this disease got its name. It's known to be due to a deletion of the short arm of chromosome 5. This is a child that has cri du chat, so you can see some canonical facies, that is the appearance of the face, hypertelorism, a slight change in the nasal bridge, a broadening of the nasal bridge. So if you read the pediatric kind of literature, that the discipline in medicine called pediatrics that takes care of small children usually, all children really, there will be kind of classical appearance of a child with Cre de Shot. So there are some morphological features. And if one looks at the karyotype, that's what we're looking at here. That's the stained chromosomes. On chromosome 5, one of the regions will be deleted at the end, the terminal deletion on at least one of the chromosomes. Okay, so that's Cre de Shot. That's a deletion. What about duplications? Well, again, duplications are the examples where during crossing over, the point where the chromosomes crossed over was inappropriate. Like usually the B's and C's would have to line up here. The CD region aligns with the BC region. Somehow there was enough DNA sequence that this happened, though it wasn't the accurate place. And the result is that this purple sister chromatid will pick up part of the blue one, and we get a duplication. So we have A on the left side, centromere, B, C, D in the normal unaffected chromatid. But the two that underwent some kind of change through this duplication, we see A, B, C, C, D. So we get a duplication of the C region here. And then as a consequence, the other chromosome loses the C. So here we have ABD missing the C. So we get a duplication in this one and a deletion here. Both could have big effects on the function of the functional output, that is, the phenotype of the organism, if these are genes that are critical. But we don't know. We have to look at these case by case because there may be little to no effect as well. Okay, so let's talk about the role that duplications has played over evolutionary time in this concept of gene families. So imagine millions of years ago in some organism, there was a single gene, served a single purpose, but then during the formation of sperm and egg, there was a duplication event, like we see here, so two C's, and that resulted in two genes. The C here is just a marker for a gene, okay? So there was a duplication that was abnormal, and those genes then get passed on to the offspring if this happened in the formation of sperm or egg. And so the parent would have one gene, the offspring would have two highly, well, identical, roughly identical genes. So now imagine that those genes have slightly different mutation rates or are in different regions of the chromosome, and one accumulates more mutations than the other. And it becomes essentially still in the same gene family, but it has slightly different functions now. So now we've got a gene family. We've gone from one gene to two, and you can imagine that could happen several more times. The best example I can think of in humans is that of the globin genes. So we've talked about globin genes in the context of sickle cell anemia so far. And remember that hemoglobin is composed of 4 different globin genes that come together to form a tetramer, and then it can take oxygen and CO2 from the tissues, and to the tissues it can take oxygen. And humans have 14 globin genes in our genome. They are believed to have arisen from a single ancestral globin gene over millions of years of biological evolution, hundreds of millions of years. You can find ancestral globin genes in very primitive organisms. We can also find myoglobin genes. Those are the ones that serve a specific purpose in muscle to bind oxygen and provide it to the muscle tissue, whereas the hemoglobins are the ones that are found in red blood cells predominantly and carry oxygen to the tissues from the lungs, and then pick up waste products and CO2 from the tissues and take that back to the lungs for off-gassing and what I'm forgetting the name of the process, but basically removing waste products as well. Okay, so why do we talk about these multiple globin genes? Because they arose through the duplication events. That's the theory, or the hypothesis really, is that there was an ancestral globin that split off to make the myoglobins, most common in muscle tissue, and the hemoglobins, which are found in like circulatory system in the blood cells, red blood cells. And further kind of differentiation or additional mutations occur, led to these different subgroups, the alpha chains and the beta chains. You can see here's beta hemoglobin, here's alpha 1 and 2. And so today we have multiple hemoglobins. They serve different purposes. So if we look at the ones surrounded in red, this one is, forgetting what that symbol is. It's a hard one. This is a sigma. This isn't lambda, but maybe it's zeta or something. These are present in embryos, so they have a different affinity for oxygen. Remember, embryonic mammals are basically getting the oxygen from the mother, and it's transferred across the placental barrier. And so the embryos have these kind of higher affinity hemoglobins that kind of can pull some of that oxygen and supply it to their own tissues. Then during development, there are new forms that are found, and then after birth, it's mostly the alpha and betas. That's what would be in all of us adults, okay? So again, this concept of gene duplication can be applied to help us understand how gene families arose over hundreds of millions of years of evolutionary time. Let's now talk about inversions. This is where the chromosome is in one orientation and part of it flips, okay? So here we see a chromosome with markers A, B, C, D, E, F, G, H, I across the entire chromosome. And so there are two different types of inversions. One could occur around the centromere, and one could occur to either side. So pericentric means around, P-E-R-I means around, and para means to the side of, okay? So again, in this case, we've got A, B, C. It should be D, E, F, G, H, I, but then the D, E, F, G is in the opposite direction that it should be. It should be facing this way, but it flipped around and inverted. Okay, so no information is going to be lost here yet. A, B, C, D, E, F, G, H, I. So if this just happened, it would be okay, probably. We could have these things called position effects because now the G is near the C versus being near the H, and that could have an effect. We'll see. But the bigger issue is that when this organism then goes to reproduce, it will have some issues when crossing over occurs between these chromosomes and others, kind of normal ones. Now, what is a position effect? I've been blabbing about it. So think of this as a chromosome. Remember, the telomeres are at the ends. We have a gene of interest here called ADE2. This is a gene that's involved in adenine biosynthesis in yeast. This is Saccharomyces cerevisiae. This is the baker's yeast. So the ADE2 gene is really important. And if it gets moved through, say, inversion or something where the green chunk ends up near the telomere, that's a different environment for that gene. Remember, the ends of chromosomes have these telomeres that are kind of tightly packed DNA that act as bumpers. So if you move a gene right next to a telomere, its expression level may go down. It could be kind of pulled into that tightened heterochromatin DNA that's not the loose stuff, it's the tight stuff. Okay, that will shut down the expression of that gene to some extent. What happens when the ADE2 gene is not functioning? It's part of a big biochemical pathway. And if it gets plugged up, that is the ADE2 gene is blocked, all the products are still formed and they're trying to go through this chemical reaction, but they build up because there's an enzyme called ADE2 that's missing, okay? And the result is that the product that was gonna get converted in this step is blocked, and it builds up and it oxidizes and turns red. So we see the red yeast here. There is something we call white sectors, so it's not a perfect blockade of the enzyme, okay? Because some of the yeast don't turn red. Scientists have also discovered this in fruit fly eyes. So there's a gene called the white gene, and it can be moved around through inversion. I think it's really susceptible to transposase mediated movement as well, but we'll get to that topic a little later in this course. So right now we're just talking about position effects, how depending on where a gene is on a chromosome could affect its expression level. We see that also with the white gene. So when the white gene is is pretty far from this packed DNA called heterochromatin. It's expressed, and the fly has these red eyes. This is okay, so this is not like the ADE2 gene. In this case, the white gene causes white eyes when it's mutated, but otherwise red eyes are the common color of a fruit fly. So now imagine the white gene gets put next to this tightly compact heterochromatin, and it's not defective, it's not mutated, it's just packed in tighter. So the expression of that gene is reduced. And the result is that sometimes it expresses okay and you get red patches in the eye. And in other cells, it doesn't express well, so you get white patches, and you get this kind of what would be called a mottled, M-O-T-T-L-E-D, mottled appearance of the eye. Okay, so we've talked about deletions, duplications. We looked at an example of how duplications lead to gene families. Then we talked about position effects, that genes can be put into different areas of chromosomes, and this might affect their function. Let's go back to inversions. We talked about it briefly, and we're not going to get too bogged down in this because it is just it's like a puzzle, okay? And so I'd like you to look at it and appreciate what's going on here. But these are called inversion heterozygotes. So now you've got a totally normal chromosome from one of the parents, and then you've got another one that, so this is the homozygote dominant copy, this is a homozygote recessive copy, all the recessive copies of the genes on this bottom one. And also, this chromosome underwent a pericentric inversion around the centromere. Now they have to pair up to do meiosis because this is an organism that is trying to make sperm or egg, I guess, okay? So now when the pairing up happens, it gets really weird because of this inversion. This chromosome is just doing its own thing, A, B, C, D, E, F, G, H, I. And this one has to conform to meet all the pairs. So look at this. You've got the D, E, F, G. So D, E, F, G. So in order to put that in the same direction, the chromosome has to flip around. And that's why we get this loop-de-loop. Now, so far, it's still going to work, right? We've got the D pairing with the D, E with E, F with F, G with G. But this is a real mess because it's going to break this stuff, and we're going to end up with all these possible outcomes. So the outer purple chromosome will be unaffected. The outer blue one would be this one. So it's kind of inner in this case. It's not affected. But the two chromosomes that pair up, they've got some problems. So again, the outer purple one, totally fine. The capital letters, the inner blue one, completely fine. All the, here it is, A, B, C, D, E. Well, it is messed up. A, B, C, and let's follow this one around. ED, okay? Because remember that one was inverted. A, B, C, ED. And so it gets messed up. Those letters don't seem to be, I must be, oh yeah, I'm saying them wrong. So A, B, C, G, F, E, D. So that makes sense, right? A, B, C, G, F, E, D. So that's the order there. A, B, C, G, F, E, D. So both of those are messed up. And as a consequence, when they resolve, you will end up with this really weird mix of alleles, right? A, B, C, D, E, the capital, and then small f, G, C, B, A. So it's all messed up. Similarly, this second from the bottom one is messed up, and then these outer ones are okay. So that's called an inversion heterozygote, and this will lead to some death of the organism. This one has almost all the genes, right? A, B, C, D. We're missing E, I think. There's an extra copy of C. So might have an effect. This one's going to definitely have an effect. It's got I, H, G, F, E, D, H, I. There's no A, B, or C genes at all. And that could be an inviable embryo, okay? Now, what happens if the inversion is not involving the centromere? We still get these weird pairings. And here we will get a totally normal outer chromosome for the purple and the blue at the bottom. But the middle ones are pretty wild, right? We can get ABC, capital, and then small d, E, A. And we can even get, in this case, because it's off center, then when those pair up, in this form, we will end up with both centromeres in the same chromosome. So this is called a dicentric bridge. So now imagine if this chromosome ends up in a sperm and it fertilizes an egg and it goes to do crossing over during recombination. You could get the different centromeres pulled in different directions. You can get all kinds of strange outcomes. The bottom line is that inversion heterozygotes, it can be okay for the first generation, but when they go to make, as long as all the genes are there, but note there could be position effects because these genes are now in totally different environments, close to a centromere near the end when it didn't used to be, like this A is at the end here, but it's at a different end. Okay. So that can create problems, and this one might not even make it because cells do things like count centromeres in order to do some of the processes that occur, like lining chromosomes up on the metaphase plate and stuff. And if there's no centromere, then it becomes a problem. So it might just get degraded. Okay, so both pericentric and paracentric inversions are problematic. We're going to end this section on how chromosomes get messed up by talking about translocations. A chromosomal translocation occurs when a segment of one chromosome becomes attached to another. Sometimes it's reciprocal, where each chromosome will give the other a bit of itself. Okay, but in all of these, it still requires that there are breaks of the DNA, that kind of strand invasion we talked about, some branch migration. So that was homologous recombination, but with translocation, sorry, I misspoke. With translocation, that can happen due to abnormal crossovers, but the ones we're envisioning are more commonly just chromosomal breaks with DNA repair, leaving things like in the wrong place. So again, there are lots of different terms thrown around. You've got simple translocations, you've got reciprocal, then we got this concept of balanced, where the pieces that are shared between chromosomes are roughly similar. Okay, let's look at this example of a chromosomal breakage with DNA repair. So you've got two chromosomes that have been duplicated, chromosome 22 and 2, and notice there's a break here now that happens. Okay, so the break happened here, and then this little chromosome with the turquoise or blue gave part of that blue chromosome to the bigger one, and the bigger one gave a little bit of this pink chromosome to the blue chromosome. So we end up with these hybrids. No genetic information is lost. It's in a different position. Okay, so that would be a common thing that could happen between two dissimilar chromosomes. We could also have inappropriate crossing over because there can be regions of homology, like small regions. Sometimes the wrong chromosomes will pair up. Instead of 1 with 1 and 7 with 7, here we get 1 with 7, and so we get a balanced reciprocal translocation. Okay, now here's an example of a balanced reciprocal or reciprocal balanced translocation. You can see that chromosome 8 has this important gene called the MYC gene. This is probably involved in things like cell cycle. This is called an immediate early gene. They're important for basic functions of cells usually. A lot of times they might be involved, as I said, in cell cycle. They could be like G protein coupled receptors kind of involved with intracellular signaling. Chromosome 14 has this region. And so notice that these kind of swapped information. And so that could affect the function of the cell due to of these chromosomes due to position effects. Let's look at another example. So just to be clear, we're talking about lots of different swaps here. So I don't expect you to memorize 2 and 22, 1 and 7, 8 and 14. This one turns out to be an important one clinically, but the one we're going to focus on is the next example. So there are a whole bunch of different disorders that affect our blood cells. We can think of things like lymphomas, leukemias. These are typically cancers that involve the cells in our bone marrow, in our blood system, like our bloodstream. So this one is one of those types of cancers. It's called chronic myelogenous leukemia, CML, okay? So it is due in 95% of the cases. CML can be traced back to what's called a translocation. It's a balanced translocation. So on chromosome 9 is this small region called the ABL. That's a gene called the ABL gene. It's a cell cycle control gene. And then on chromosome 22, it's a smaller chromosome. There's a region called the BCR. This is referred to as the breakpoint cluster region. Okay. So I think it's an area that can more easily break. And so we get this translocation. And it results in this funny little chromosome that has most of chromosome 22, still got the breakpoint cluster region, the green thing, but it picks up the end of chromosome 9, including that Abel gene, and it just puts it right next to the BCR. It turns out, so scientists knew about these chromosomes in the 60s. They were looking, clinicians would look at blood smears of patients that had these leukemias, and they started saying, well, they all have this kind of different chromosome called the Philadelphia chromosome. I left out a picture, but the scientist who discovered it was working in Philadelphia. In any case, it's associated with this CML in a high percentage of the cases. And there's a way that clinician scientists or geneticists kind of write this translocation between 9 and 22 at this region. So the Q34.1 region is swapped with the Q11.2 region. You don't need to memorize that. That's just like the arcana. That's how geneticists, clinical geneticists write out the genotype kind of, or really it's not as much the genotype. It's sort of getting at that, but it's talking about the changes in the chromosome structure. Okay. All right. So what happens? Well, the Abel gene, as I mentioned, is involved in things like the cell cycle, and it's a tyrosine kinase. So its job is to phosphorylate other proteins in a signal transduction cascade to either accelerate the division of cells or to slow it down. And what happens that goes wrong here is when the Abel gene is put next to the BCR gene, it just turns it on full blast. So instead of there being regulation of the expression of that gene, it is now just fully on, okay, constitutively expressed at a high level. And that leads to cells, certain types of cells, these myelogenous cells, growing out of control. And essentially that causes cancer. Now, there was a breakthrough in clinical therapeutics for cancers in the mid to late 1990s. This was a multi-team effort, scientists at universities and also several big drug companies. And it led to the discovery, it might have been one big drug company, I'm forgetting if it was Pfizer or Roche or somebody, but it led to there it is right there, Novartis. Okay, my own notes. So this drug company called Novartis developed a blocker of this particular tyrosine kinase, the Abel gene. So they developed a small molecule that could be taken as a drug. I'm not sure if it can be taken orally, but it's a tyrosine kinase inhibitor. So they could treat these types of cells that are growing out of control by kind of shutting them down with this small drug molecule. It has been quite a success. And so just in summary, then, we've looked at deficiencies, deletions of chromosomes. We talked about Cre de Shot. We looked at duplications and how that can happen during meiosis, and it can also lead to gene families. We looked at inversions, how those can really mess up chromosomes by swapping the orientation from like inline to the opposite direction. We also, in this, talked about how when genes are put into different positions on chromosomes, this can fundamentally affect their function. And that's called a position effect. And then we ended up this discussion with this concept of translocations, how part of one chromosome can go to another in a simple translocation, or in a reciprocal translocation, two chromosomes give each other parts. And we talked about how this could happen as an inappropriate response in recombination, like mitotic recombination, or it could just be due to breakage and repair of chromosomes. And the example we looked at there was this last example of chronic myelogenous leukemia, which is due to a translocation between chromosomes 9 and 22. Okay, so the last topic we're going to discuss today is important for anyone interested in the immune system, which everybody should be interested in it because it's our protection from invaders like viruses and parasites and bacteria, right? So most of you probably know about antibodies coming out of the COVID pandemic here. Antibodies are these proteins that are in our blood cells. They're floating around in our bloodstream that are able to bind to things that we recognize generally as not being part of ourselves. And so there's this structure to it. It kind of looks like a Y. It's composed of 4 different polypeptides, 2 heavy chain, the blue things, and 2 light chain, the red pink things. And those are each coded for by genes. There are 5 different types of major antibodies in humans. You'll learn about that in immunology. We're just going to look at them generally here. And they're made by B cells in our bone marrow. Okay. So they bind to antigens. That antigens could be invaders from outside our body. They could be viral particles. They could be bacteria or other types of parasites. Occasionally, in a condition called autoimmune immunity, and there are lots of, it's not one condition, there are lots of types of autoimmune responses. Our own body attacks itself, and sometimes antibodies are involved in that. Now, so think about this. There's an infinite number of antigens. Antigens can include small molecules. People can develop antibodies to drugs. In fact, there's a big push right now, and this works was going on at the U of M, but the scientists moved to Seattle to generate antibodies against like drugs that are oftentimes used by humans for recreation, but that people become sort of develop some kind of substance use disorder. So things like fentanyl, things like cocaine, people are trying to develop antibodies against those so a person could become immunized against those. But mostly we think of antibodies as being a natural thing that happens that we develop in response to a challenge by things like different bacteria or viruses or parasites. So that's getting at the last point on this page. We need access to billions of different antibody combinations. But if you just, even if we had 4 separate genes, you can't get enough combinations of those from the number of genes we have. Okay, we only have 22,000 genes. Just a handful of those are devoted to making antibodies, the globin genes. Sorry, the immunoglobulin genes, not globin, that's a different class. But immunoglobulins make antibodies, and we have a limited number of those genes. So even if they could all, in every combination, mix and match, it would not account for all of the different antibodies we can generate as part of our immune response to protect our bodies. Now, let's just do a quick overview of the immune system. So there are 3 major types of immunity that humans and mammals have in general. Innate immunity and adaptive are the 2 big categories. But before that, we've got the barrier function. That would be our skin, the tears, saliva, the skin oils, everything that is protecting our skin. We call that the barrier function. That's like our first line of defense, right? It includes the saliva, so we might take things in, but we may cough it up and spit it out. That's all barrier function. Hopefully that's intact. But imagine a scenario where you have a slight cut or where the barrier isn't enough to keep the thing from getting into your body. Then we have the first level of defense. Well, it's not really first or second. So the first level is the barrier function, but once something is inside of you, then you have 2 other ways to deal with the invading microbe or virus. One of them is innate immunity. Those are the cells that we have that are always kind of looking around, surveilling our bloodstream and our tissues for things that shouldn't be there. Things like macrophages, natural killer cells, neutrophils, eosinophils, and basophils. These 3 are all in blood. These can leave the blood. So like macrophages can cruise around tissue and maybe they're looking for a bacterium. Maybe they go to the site of an infection. Natural killer cells can inactivate bacteria on their own with a series of things like complement proteins. So that's kind of, and the neutrophils, eosinophils, and basophils are the types of white blood cells we have. So if you've ever been sick and gone to the doctor, they take your blood, they may say, well, your white cell count is high. That's because these innate immune cells have kind of launched into action and proliferated, and they are there to kind of remove the invading bacteria or viruses. Then we have adaptive immunity, and there are some cells that kind of are at the interface, like T cells and natural killer T cells. The adaptive immune response is the one that kind of can learn from the current situation. And so, for instance, these B cells will, so it's complicated, but imagine that a new bacterium shows up on the scene and it's in some of your tissues and it's swollen and warm. And so the innate immunity is trying to do its job, but it's getting kind of overwhelmed. And so the body can also call on these other mechanisms like adaptive immunity. And so these cells will chop up the antigens, the proteins on the surface of the invading bacteria, and it will present those to a B cell. So there are these cells called APCs, antigen presenting cells. I believe they can be like T cells, and they give the B cell a little bit of information. They say, this is what this invading bacterium looks like in part of its region. And then the B cells can generate specific antibodies, that is this antigen binding site, to know specifically where to bind on that bacteria. And once the B cells produce a lot of those antibodies, then those can coat the invading bacterium, for instance, and then some of these other cells can come in and destroy it and remove it from the body. The B cells have a memory, sometimes a lifetime of memory. So if you are given an immunization, that is the antigen, and the B cells make antibodies for those. Now, your B cells will initially make a certain amount of antibodies, and then when they see the antigen a second time, they'll make a lot more. And that's the basis for the booster shot. Some types of immunity persist for lifetimes. So for instance, some vaccines we get, we don't need boosters later in life. I think like the hepatitis A vaccine gives pretty lasting immunity. Things like flu, they're constantly changing. So influenza, we get a new vaccine every year. And it seems like something like COVID might be similar to that. Another example is measles. Many of us were vaccinated for measles decades ago. Hopefully we have enough immunity. But sometimes you need a booster, like a tetanus shot. You need a booster every 10 years or so. So those B cells can persist, but then with some of them, you need to kind of re-up and give it more information to stimulate the production of new antibodies. So we need lots of antibodies. We don't have enough genes to code for the billions of antibodies we need. This is where genetics comes in. And this is a thing called somatic recombination. It's really a type of programmed gene rearrangement in the DNA, okay? But it's not meiosis. It's not in the formation of sperm and egg. This is in a functionally like adult organism. I mean, it could happen in childhood too, but it's a mature kind of system that then rearranges its genes for these B cells to make antibodies that are highly variable. So what we're going to be thinking about is kind of these regions. We're going to look at light chain. It happens with heavy chain as well. We're going to focus on one. And this is a concept called VDJ recombination. So there are multiple genes in a clusters in our genome that code for the immunoglobulins. And so what we're going to look at here is VDJ recombination. There are certain areas that have a bunch of modules, little areas that code for polypeptides that we call the variable region, and then the D region and the J region, okay? Now, I've linked to some resources here. This is a good video to watch. This whole process was discovered by this scientist shown here named Susumu Tanagawa. In the 1970s, he was a young scientist at MIT in Cambridge, Massachusetts, and he discovered the process by which somatic recombination occurs in the formation of these antibodies to give billions, if not more, potential combinations so that we can have antibodies to basically anything that we're confronted with. For this work, he received the Nobel Prize in 1987. And I really like to talk about Susumu Tanagawa because, one, he's a brilliant person. I don't know him personally, but he shifted gears. After he made this extremely important discovery in the immune system, he became a neuroscientist, and he is one of the world's premier kind of top neuroscientists studying how the brain functions, how connections are formed in the brain. So a good lesson about work life, that you may train in one thing, you might even make great discoveries in one area, and you may end up doing a totally different thing with another part of your life. And that's totally okay. All right, so we should get into the science. What actually happens here? Okay, so again, not to get too, we're going to go into detail, and one of the videos goes into even more detail. And then I encourage you to take immunology if this excites you or you're interested in this. And I believe you'll go into even a little more detail in the immunology class. But I have to go back and just say, okay, we've got these chunks of antibodies that are called the light chain. They're the small pieces. And then the major piece in the middle is called the heavy chain. And every antibody has two heavy chains and two light chains. They are produced by separate genes. The two heavy chain ones would be the same gene, and the light chains would be the same gene here for this particular antibody. And then after they are formed, they form these disulfide bonds that bring them together to form what we would call the quaternary structure. And then at the tips of these, that's the antigen binding site. So you can see the antigen here. It has a specific fit. You could think of it as kind of a lock and key. It's not the most accurate, but if that helps you understand it, there's like a slot that the antigen goes into, and then that kind of activates that antibody now has the information. And this antibody might be present just floating around, or it may be attached to a cell. But after it binds this, generally a cell would then attach that antibody and kind of remove it. So it's a way to kind of take this antigen out of commission. Okay, so we're going to look just at what happens with the light chain recombination. Heavy chain can do it as well, but we're going to focus on this light chain. Okay, so somewhere in your genome is this cluster of different modules, and there's going to be the L region, the linker region, the variable region, the joining region, and the constant region. Okay, so what happens? This is germline DNA. That would be like sperm and egg. This is DNA in the B cell. Okay, so recombination, somatic recombination during B cell maturation. As the organism is growing, the B cells start to form and they're developing. And there will be a little bit of recombination that happens there, okay? And so that's somatic recombination. As you notice here, the V2 region pairs up with the J3 region, and everything in the middle is lost. So LN, VN, J1, J2 just goes away. That DNA gets degraded. So this particular B cell now just has a limited number of the variable regions. It just has picked one of them. And so then it's got a bunch of joining regions and a constant region. So now it's transcribed, okay, from the L2 promoter. That's this P here, the L2 region. And once that's transcribed, then some other stuff happens. So the internal sites get spliced between the J3 and the constant region. So now we're down to the L2, V2, J3, and C. That gets translated into protein. The leader sequence, the L2, is removed. So we have a variable region, a joining region, and a constant region. Let's go back and look at the light chain a couple slides back. We've got the variable region, hypervariable. I think that's equivalent to joining, or it's close to that. And then we have the constant. So those three regions were formed here. But any of these combinations could happen. And so what we see here is these LV segments, these leader and variable segments. There are, depending on the particular, there are 5 different immunoglobulins with 5 different light chain genes, gene regions, and those could have between 70 and 100 LV segments. Here we're just seeing the first 2, and then they put the N, meaning kind of, it could be a big number. And so imagine the combinations. We could have 100, and to do that math, we do the product rule. So we've got 100 times 5 times 1. So we could have 500 different outcomes here. And that's not including the fact that the hypervariable region can mutate and cause other mutations. And this is just for one of the 5 classes of antibodies, okay? So I introduced this as VDJ recombination. There is no D region in the light chain genes, and the examples I could find were for light chain recombination. But the point is that there's this, imagine you have all of these different like cards, and you need to pick 3 out of like 20 decks of cards, okay? And you could pick 3 from any of the decks of cards and mix them together to get your hand. That's how antibody somatic recombination is. You start with this palette of all these different variable joining and constant regions, and depending on how you link them up, you get a very different outcome. And that leads to the diversity in this region that binds antibodies so that the cell can basically, it doesn't do this in a thoughtful way. It's just generating antibodies. And then if the antigen fits, it then kind of amplifies it. So then the B cell gets activated and it makes tons of these antibodies. And then those B cells that have these specific antibodies, they lay dormant, so to speak, in our bone marrow. And if they are challenged again in the future with the same antigen, then they ramp up production and have much higher levels of antibody production. So the basis for immunization, getting a vaccine, is that initially that vaccine finds an antibody and it activates the B cells. And then you do the booster and it makes lots more B cells. And then some of those are like memory cells. They can stay in the bone marrow for long periods of time, maybe even a lifetime. And then if you ever see that antigen again, if you're infected with the same bacterium or virus, then your body can mount an adaptive immune response along with the innate immune response to try to clear out those pathogens, okay? The next picture just shows another example of somatic recombination for light chain antibodies. Here again, we've got 300 variable regions. We got 4 joining and 1 constant. And so initially the variable region links up with a joint region, and that's in the somatic recombination in the DNA. And then that gets expressed and we get some splicing, and ultimately we get a protein that ends up in the antibody with, in this case, it was the variable region 78, the joining region 2, and the constant region. So that's one of the many possibilities that you could get out of just joining one variable with one joining region here. And that's called site-specific recombination. It happens in non-germline cells, right? So it's happening in the B cells. We can see that back here. It says somatic recombination during B cell maturation. This is just what the original sperm or egg sequence would look like, but it gets recombined in the formation of these B cells. That is the end of these topics. I'm going to go back to the very first slide and just let's highlight what we did here today in this class. So we talked about recombination in general. We talked about the holiday model to explain recombination as it occurs in meiosis primarily. Then we talked about ways that chromosome structure can be altered. This could happen during recombination, or it could happen during mitosis or another time in the cell cycle. And we ended it up with a special type of recombination that happens in somatic cells, so not in the germline, in particular in B cells, to form a vast repertoire of different antibodies that have different antigen binding capabilities. Thank you for your attention.